Hello.
We are excited to introduce a brand new audio research book where we will read articles for you. Our first article will focus on the cryo-electron microscope structure of tau filaments from Alzheimer's patients. Here is the title of the article: Cryo-EM Structures of Tau Filaments from Alzheimer's Disease. This paper was published in Nature in 2017 and conducted at the MRC laboratory in the UK. Let's begin our discussion of this groundbreaking research.
Alzheimer's disease, AD, is the most common neurodegenerative disease, and there are no mechanism-based therapies. AD is defined by the presence of abundant neurofibrillary lesions and neuritic plaques in cerebral cortex. Neurofibrillary lesions are made of paired helical and straight tau filaments, PHFs and SFs, whereas tau filaments with different morphologies characterize other neurodegenerative diseases. No high-resolution structures of tau filaments are available. Here we present cryo-electron microscopy, cryo-EM, maps at 3.4 to 3.5 angstrom resolution and corresponding atomic models of PHFs and SFs from AD brain. Filament cores are made of two identical protofilaments comprising residues 306 to 378 of tau, which adopt a combined cross-beta-beta helix structure and define the seed for tau aggregation. PHFs and SFs differ in their inter-protofilament packing, showing that they are ultrastructural polymorphs. These findings demonstrate that cryo-EM allows atomic characterization of amyloid filaments from patient-derived material and pave the way to study a range of neurodegenerative diseases. Main neurofibrillary lesions correlate strongly with cognitive deficits, making them an important therapeutic target for Alzheimer's disease. Dominantly inherited mutations in MAPT, which encodes tau protein, cause frontotemporal dementia and Parkinsonism linked to chromosome 17, FTDP17T, showing that dysfunction of tau is sufficient to cause neurodegeneration and dementia. Ultrastructurally, tau inclusions are made of paired helical filaments, PHFs, and straight filaments, SFs. Negative stain electron microscopy shows that the cores of PHFs and SFs consist of a double helical stack of C-shaped subunits, whereas the amino and carboxy terminal regions of tau are disordered and project away from the core to form the fuzzy coat. In Alzheimer's disease, tau filaments form from full-length tau in cells, but have lost most of their fuzzy coat in extracellular ghost tangles. They have a cross-V structure. Neurofibrillary lesions appear to propagate in the course of Alzheimer's disease through connected pathways, from subcortical areas to the transentorhinal cortex, limbic system, and neocortex. Although the molecular tau species responsible for neurodegeneration are unknown, short filaments are the main species of seed-competent tau in the brains of transgenic mice, expressing human P301S tau-10. 6 tau isoforms ranging from 352 to 441 amino acids in length are expressed in the adult human brain. These are produced by alternative mRNA splicing of transcripts from MAPT. They differ in the presence or absence of inserts of 29 or 58 amino acids in the N-terminal half, and the inclusion or absence of the 31 amino acid microtubule binding repeat encoded by exon 10 of MAPT in the C-terminal half. Inclusion of exon 10 results in the production of 3 tau isoforms with 4 repeats each, 4R, and its exclusion in a further 3 isoforms with 3 repeats each, 3R. The 4 repeats, R1, R4, comprise residues 244 to 368 in the 441 amino acid tau isoform. Neurodegenerative diseases with abundant filamentous tau inclusions are referred to as tauopathies. Besides Alzheimer's disease, these diseases include tangle-only dementia, chronic traumatic encephalopathy, argyrophilic grain disease, progressive supranuclear palsy, corticobasal degeneration, globular glial tauopathy, and Pick's disease. Unlike Alzheimer's disease, these other diseases lack amyloid beta plaques. In AGD, PSP, CBD, and GGT, only 4 R-tau isoforms are found, whereas in PID, only 3 R-tau inclusions are present. The existence of human tauopathies with distinct filament morphologies has led to the suggestion that different molecular conformers of aggregated tau may exist. Multiple molecular conformers may also explain why AD brain-derived filaments are more effective than in vitro assembled filaments of recombinant protein in inducing tau pathology in mouse brain. Progress in understanding tauopathies is hampered by the lack of atomic structures of tau filaments from brain. Studies of amyloids are complicated by potential differences in structure between filaments in the brain and those assembled in vitro. Low-resolution cryo-EM structures are available of in vitro assembled filaments of beta-amyloid, and solid-state NMR has been used to study the structures of recombinant amyloid beta and alpha-synuclein. Even when seeding in vitro filament growth with extracts from diseased brain, structures may not be amplified according to their relative abundance in the brain. Here we present atomic models as determined by cryo-EM for the core of PHFs and SFs that were purified from AD brain. Neuropathological characteristics. We used cerebral cortex, figure 1A, of a 74-year-old female with a neuropathologically confirmed diagnosis of AD. The interval between clinical diagnosis and death was 10 years. The patient's apolipoprotein E genotype was epsilon 3 by epsilon 4. Exons 16 and 17 of the amyloid precursor protein gene, exons 3 to 13 of the presenilin 1 gene, as well as exons 4, 5, 7, and 12 of the presenilin 2 gene, including adjacent intronic sequences, were sequenced and no disease-causing mutations were found. The patient's mother had died aged 86 with a 16 years history of AD. Thioflavin S staining showed the presence of abundant neurofibrillary tangles and neuritic plaques in cerebral cortex. Figure 1b. The sarcosyl-insoluble fraction of the patient's neocortex contained numerous PHFs and SFs, which were composed of full-length hyperphosphorylated tau as assessed by immunogold EM, extended data figure 1. Negatively stained PHFs had a longitudinal spacing between crossovers of 650-800 angstrom and a width of about 300 angstrom at the widest part and 100-150 angstrom at the narrowest. SFs were about 150 angstrom wide with crossover distances ranging from 700-900 angstrom, figure 1c. By Western blotting, pathological tau bands of 60, 64, 68, and 72 kilodalton were present, extended data figure 1a. These characteristics are like those described previously for PHFs and SFs from AD brains. Sarcosyl-insoluble material seeded aggregation of full-length human tau in cultured cells. Figure 1 shows structure of tau filaments from Alzheimer's brain. A coronal section at the level of the precentral gyrus of the brain used in this study shows an enlarged lateral ventricle, gray background. Gray matter from frontal cortex, red, and temporal cortex, magenta, was used for cryo-EM. B. Thioflavin S, staining light microscopy showing abundant neurofibrillary tangles, yellow arrows, and a neuritic plaque, white arrow, in temporal cortex. C. Negatively stained electron micrograph of purified tau filaments, in which PHFs, blue arrows, and SFs, green arrows, are readily distinguished. DE cryo-EM reconstructions of PHFs, blue, and SFs, green. PHFs and SFs are ultrastructural polymorphs. The sarcosyl-insoluble fraction of the patient's cerebral cortex was purified by differential centrifugation and gel filtration. Methods was purified with no disruption to the structures of PHFs and filtration. Extended data figure with no disruption to the structures of purified sarcosyl-insoluble fraction also seeded the aggregation of full-length human tau in cultured cells. Extended data figure 2D. The two types of filaments could be readily distinguished in cryo-EM images, and corresponding 3D reconstructions were calculated separately to overall resolutions of 3.4 and 3.5 angstrom. Extended data figure 3 using helical reconstruction in Reliant. The maps showed clear side chain densities, figure 2, and beta strands were well separated in the direction along the helical axis, figure 1e. Allowing us to propose stereochemically refined atomic models for the cores of PHFs and SFs. Methods. Consistent with previous findings, both PHFs and SFs are composed of two protofilaments with C-shaped subunits. Successive rungs of beta strands along a protofilament are related by helical symmetry, with a rise of 4.7 angstrom and a twist of approximately 1 degree in both PHFs and SFs. In PHFs but not SFs, the two protofilaments are related by an approximate 2 sub-1 screw symmetry. A clear pattern of side chain densities and various landmark features in the two maps revealed that the core of both protofilaments in PHFs and in SFs is composed of residues V306 to F378, that is all of R3 and R4, as well as 10 amino acids carboxy terminal to the repeats, extended data figure 4. The protofilament cores of PHFs and SFs are similar, extended data figure 5, showing that they are ultrastructural polymorphs, reminiscent of fibrils formed by a peptide of transthyretin. Additionally, weaker densities appear at both the amino and carboxy terminal regions of the core. Orange density in figure 2. The weaker densities suggest that these regions correspond to a mixture of peptides and/or a more dynamic or transiently occupied structure than the filament core. The weaker density at the amino terminal region can accommodate approximately 16 residues in a beta sheet-like conformation that may represent a mixture of the C termini of R1 and R2, which would result from the 3R and 4R alternative splicing products. This is consistent with our Western blotting results, which indicated that all 6 brain tau isoforms were present in PHFs and SFs, extended data figure 1A. Another region of additional density is interacting with the side chains of K317, T319, and K321 in both types of filaments, red arrows in figure 2. This density is strongest in SFs, where it forms part of the protofilament interface, whereas in PHFs, it occurs at the surface of both protofilaments. Figure 2 shows cross-sections of the PHF and SF cryo-EM structures, cryo-EM density and atomic models of PHFs, A, and SFs, B. Overviews of the helical reconstructions, left, show the orientation of the cross-sectional densities, right. Sharpened, high-resolution maps are shown in blue, PHFs, and green, SFs. Red arrows point at additional densities in contact with K317 and K321. Unsharpened, 4.5 angstrom low-pass filtered density is shown in gray. Unsharpened density, highlighted with an orange background, is reminiscent of a less ordered beta sheet and could accommodate an additional 16 amino acids, which would correspond to a mixture of residues 259, 274, R1, from 3R tau and residues 290, 305, R2, from 4R tau. Generality of PHF and SF structures. We used pronase treatment, mass spectrometry, and antibody labeling to corroborate our model of a structured R3 to R4 core with extending densities at the amino and carboxy termini. Pronase treatment has previously been shown to remove the fuzzy coat of PHFs and SFs, while leaving the structured core intact. We confirmed that cryo-EM structures of the core of both PHFs and SFs do not change upon pronase treatment. Extended data figure 6. Mass spectrometry of the pronase-treated filaments showed that residues I260 to R406 of 3R and residues K281 to R406 of 4R tau are present in the structured core of the filaments, with R3, R4, V306 to R406 being by far the most abundant species, extended data figure 7A. This finding is also in good agreement with peptides identified in earlier work, and a recent report on mass spectrometry of trypsin-treated PHFs and SFs from 10 AD cases. In addition, to confirm that R2 is not part of the pronase-resistant core, we used anti-4R, an antibody raised against V275C291 of R2, with D279. In agreement with a pronase-resistant R3, R4 core, anti-4R detected both PHFs and SFs before, but not after pronase treatment. By contrast, antibody MN423, which recognizes tau truncated at E391, detected PHFs and SFs only after pronase treatment. Extended data figure 7B, as reported previously. The model of an ordered R3 to R4 core is also consistent with the observation that antibodies BR135 and tau C4, which recognize sequences inside R3 and R4 respectively, do not label either PHFs or SFs, whereas they do label PHF tau on denaturing gels, extended data figure 1A. Sequence analysis of the PHF core also showed the presence of R3 to R4 of tau. Thus, we propose that PHFs and SFs have a common core structure composed primarily of R3, R4, with a mixture of R1 and R2 directly preceding V306. Structure of the common protofilament. The core of PHFs and SFs is composed of 8 beta sheets, beta 1,8, that run along the length of the protofilament, adopting a C-shaped architecture. Each C is comprised of a beta helix region, where 3 beta sheets are arranged in a triangular fashion, and 2 regions with a cross-beta architecture, where pairs of beta sheets pack anti-parallel to each other. Figure 3AB. Forming such a continuous beta-strand architecture along the chain of an individual tau molecule, 80% of residues are in a beta-strand conformation occupying only 13.3% of the total phi-psi space, is achieved by interspersing the beta-strand regions with beta-breaking prolines, P312, P332, P364, beta-turn glycines, G323, G355, or beta-arc residues, E342, D348. The strain of having many twisted beta-strand regions is further relieved by changes in the height of the chain along the helical axis, resulting in differences of up to 10 angstrom between the highest point in beta 1 and the lowest point in the tip of the beta helix, figure 3C. Thereby, an ordered in-register parallel hydrogen bonding pattern is maintained throughout the protofilament structure. Additional hydrogen bonds that stabilize the protofilament are implied by the side chains of ladders of asparagines and glutamines. Starting from the termini, figure 3D, there is a heterotypic cross-beta interface formed between beta 1 to beta 2 and beta 8. The amino terminal end of the ordered core is formed by the hexapeptide 306 VQI-VYK 311, which forms a complementary packing interface with residues 374 to 378 from the opposing beta 8 by face-to-face packing of hydrophobic groups. Strands beta 2 and beta 8 pack against each other through a polar zipper motif. Next, there is a right-angle turn accomplished through G323 and G326 on one side and a compact 364 PGGG 367 motif on the other side. It is possible that cysteine C322 forms intrasheet disulfide bonds, but we were unable to detect a regular pattern that would allow us to visualize density for such bonds in our helical reconstruction. A hydrophobic cluster of L324, I326, and V363 stabilizes the region immediately following the turn, and the cross-beta interface between beta 3 and beta 7 is further cemented by hydrogen bonds between the side chains of H328 and T361. Following beta 3, residues 332 PGEG 335 adopt an extended beta spiral conformation. Finally, the two sides meet through a beta helix structure that is defined by 3 beta strands in R4, beta 4 to beta 6. 2 residue, E342, K343, and 3 residue, 347 KDR 349, beta arc corners punctuate the triangular beta helix geometry, which is closed with a pivotal approximately 70-degree glycine conformation, G355, hydrophobic clustering, aliphatic stacking, V339, L344, V350, and I354, and aromatic stacking, F346, stabilize the interior of the beta helix. Alternating charged residues between E338 and R349 on the solvent-exposed surface of the beta helix provide intramolecular charge compensation that offsets the repulsive effects of stacking side chains with identical charge. Figure 3 shows the common protofilament core. A. Sequence alignment of the 4 microtubule binding repeats, R1 to R4, with the observed 8 B-strand regions colored from blue to red. The 16 residues from R1 or R2 that may form an additional less ordered beta sheet are indicated with gray dashed lines. B. Rendered view of the secondary structure elements in 3 successive rungs. C, as in B but in a view perpendicular to the helical axis, revealing the differences in height along the helical axis within a single molecule. D. Schematic view of the protofilament core. PHFs and SFs form different interfaces. The ultrastructural polymorphism between PHFs and SFs is due to differences in lateral contacts between the 2 protofilaments. In PHFs, the 2 protofilaments form identical structures that are related by helical symmetry, and the interface is formed by the anti-parallel stacking of residues 332 PGGQ 336, figure 4AA. The offset of half the beta strand distance along the helical axis results in a staggered packing of 333 GGG 335, from both protofilaments in PHFs. In particular, the glycine tripeptide adopts a characteristic polyglycine 2 beta spiral structure that forms a hydrogen bonding pattern both within and between the two protofilaments. The PHF interface is further stabilized by the formation of two hydrogen bonds between Q336 and the backbone carboxyl of K331 on the opposite protofilament. In SFs, the two protofilaments pack asymmetrically, figure 2b. The weaker beta sheet-like density at the amino-terminal end of the structured core is only visible in one of the protofilaments, as steric hindrance at the protofilament interface prevents the formation of a peripheral beta strand on the second protofilament. Although the beta helices of the two protofilaments appear to be in a similar chemical environment, the beta helix of the second protofilament is more disordered. The backbones of the two protofilaments are nearest each other between residues 321 kcgs, 324 of the first, and 313 Velsk 317 of the second protofilament, where the protofilaments are packed against each other at approximately the same position along the helical axis, figure 4b. However, these residues do not form interprotofilament salt bridges or hydrogen bonds, nor does the interaction seem to be mediated by hydrophobic packing. Instead, the SF protofilament interface appears to be stabilized through the region of additional density that is interacting with the side chains of K317, T319, and K321 of both protofilaments. Red arrow, figure 2b. These residues are part of the epitope of the ALZ50 and MC1 antibodies that is formed by two discontinuous portions of tau, 7-EFE9 and 313 VDLS-KVTS-KC322. Nuclear magnetic resonance (NMR) experiments on heparin-induced filaments of recombinant 4R tau confirm the interaction between the amino terminus and residues 313 to 322 of the structured core, which is strongly affected by ionic strength and takes place mainly through intramolecular interactions. Therefore, although we cannot preclude the role of other polyanionic molecules, we speculate that the additional density corresponds to 7-EFE9, and that salt bridges between the negatively charged glutamates of the amino terminus and the positively charged lysines of the core stabilize the interaction between the two protofilaments. Similar additional density is also interacting with K317 and K321 in PHFs, where it does not contribute to the protofilament interface. Red arrows, figure 2a. Mutations directly preceding the 7-EFE9 motif, that is R5L and R5H, have been observed in FTDP17T. Figure 4 shows protofilament interface in PHFs and SFs. A. Packing between residues 332, PGGQ 336 of the two protofilaments in PHFs. Interprotofilament hydrogen bonds are shown in orange and green. Intraprotofilament hydrogen bonds are shown in yellow. B. Packing between residues 317 to 324 and residues 312 to 321 of the protofilaments in SFs. Additional density between the side chains of lysines 317 and 321 is shown as a green mesh. Discussion. ADPHFs and SFs consist of an ordered core of pairs of protofilaments comprising residues 306 to 378 and disordered amino and carboxy termini forming the fuzzy coat, figure 5. The core forms a sufficient platform for the incorporation of both 3R and 4R tau into the growing filament. As we were unable to detect any order in the fuzzy coat, this heteromolecular addition may occur in a random manner. This would explain why PHFs and SFs are able to seed in vitro aggregation of recombinant samples containing only 3R tau, only 4R tau, or a mixture of 3R and 4R tau. It may also explain why the intracerebral injection of PHFs and SFs from AD brain leads to neuronal uptake, seeding and propagation in wild-type mice, since the tau 306 to 378 sequence is identical in humans and mice. The ordered core is probably necessary and sufficient for the seeded assembly of tau in AD brain. The protofilament core is composed of a combination of two structural motifs that are closely associated with amyloid and prion structures, cross-beta packing, and beta helices. This structure is markedly different from the conformation of tau bound to microtubules, where residues 269 to 284 and 300 to 312 adopt a hairpin conformation, and the rest of the structure appears disordered by NMR. The generic cross-beta structure is widely accessible to peptides of varying lengths with unrelated amino acid sequences. In AD-tau, in-register anti-parallel beta sheet packing is mediated through a mixture of hydrophobic and polar interactions. The amino-terminal part of the cross-beta structure is formed by the hexapeptide 306-VQIVYK311, which is essential for the assembly of tau filaments. In contrast to homotypic interactions seen in microcrystals of isolated peptides, the hexapeptide in AD-tau packs through a heterotypic, non-staggered interface with the opposing residues 373 to 378. The same packing interface is absent in the widely used K18 and K19 constructure, which span 3 or 4 repeat domains of recombinantly expressed tau and terminate at E372. Therefore, filaments made of K18 and K19 proteins cannot represent the complete core structure of AD brain-derived PHFs and SFs, and care should be taken when extrapolating experimental results derived from the use of constructs ending before F378 to tau aggregation in vivo. In contrast to generic cross-beta structures, beta helices are mediated by specific interactions that are not accessible to most amino acid sequences. Beta helices can only arise from sequences with a specific pattern of hydrophobic residues on the inside, polar residues on the outside, and a pivotal beta arch forming glycine at the end of the motif. An example of such beta helices is the prion-forming domain of HETs from the filamentous fungus Podospora anserina. The HETs prions form a beta-solenoid core, which is essential for infectivity, composed of 2 charge-complementary left-handed beta-helical windings and a linker region. Despite having little sequence similarity, the beta helices in AD-tau and HETs 218 to 289 PDB entry 2 RNM adopt a similar fold, extended data figure 8, raising the possibility that this motif may serve as a scaffold for prion-like templated misfolding. The beta helix in tau is closed by G355, which is located in the 353 Kigs 356 motif of R4 that comprises the potential phosphorylation site S356. Knowledge of the atomic coordinates of tau filaments may be useful for the rational design of specific inhibitors of tau aggregation, as well as tracer compounds. Luminescent conjugated oligothiophonies, LCOs, bind to HETs 218 to 289 through exposed lysine ladders on the surface of the beta solenoid. Therefore, LCOs such as pentameric formal thiophene acetic acid, PFTAA, a fluorescent dye that identifies neurofibrillary lesions in AD brain sections, may bind to aggregated tau in a similar way. The specific structure of the beta helix, or possibly the protofilament interface, as opposed to the more generic cross-beta structure near the termini of the core, may also provide a binding site for positron emission tomography ligands that show specific binding to AD-tau filaments, but not to tau filaments from non-AD diseases, beta-amyloid, or alpha-synuclein. Different molecular conformers of tau have been suggested to be related to specific neurodegenerative diseases. The structures presented here show how different protofilament packing interactions lead to ultrastructural polymorphism in tau filaments. Protein sequencing and mass spectrometric analyses of tau filaments from various diseases have indicated that in PID, CBD, and PSP, R1 and R2 are more resistant to trypsin digestion than in AD. This raises the possibility of another type of polymorphism, where tau protofilaments may form from alternative combinations of the repeats, R1, R2, and R2, R3 from 4R tau, and R1, R3 from 3R tau. Extended data figure 9. Whether these hypothetical conformations exist in other tauopathies remains to be seen. Figure 5 shows schematic representation of full-length tau filaments. A shows primary structure of the longest tau isoform in human brain. The 2 inserts near the amino terminus are labeled N1 and N2. The microtubule binding repeats are labeled R14. The 7EFE9 motif near the amino terminus is marked with a blue circle. B shows the 6 different isoforms pack randomly along the helical axis. C shows, in PHFs, the amino terminal region of tau monomers in both protofilaments folds back to form an additional, less ordered B sheet against beta 1 to beta 2 of the ordered core, then becomes disordered, and folds back to form an interaction between 7EFE9 and lysines 317 and 321 of the core. D shows in SFs, only a single additional beta sheet can form, and the amino terminus of one of the protofilaments forms. Conclusion. The cryo-EM structures of ADPHFs and SFs establish a basis for understanding the differences between molecular conformers of tau aggregates, and explain how different isoforms are incorporated into the filaments. Our results demonstrate that amyloid structures from human brain may be obtained by cryo-EM. This opens up new possibilities for studying the molecular mechanisms underlying a wide range of neurodegenerative diseases.
Thank you for joining us today. Stay tuned for more articles, and please let us know if you find this format engaging and informative. Your feedback is highly appreciated.